I begin my lecture, a bit of an advertisement, so let me take just 15 seconds to talk about that.
To turn on your metal.
To turn on the metal. Press the middle button. Right, I've seen Infinity War, and it turns out badly for everyone. Right, so actually the ecology is very bad in Infinity War, just that's an aside since I'm wearing a metal. You know, it only takes one generation to recover from whatever that person did. So this is a PhD in quantitative biosciences. I'm the founding director. It's based within the College of Sciences, Georgia Tech. I know most of you are already graduate students doing PhDs. If you're a master's student or if you know undergrads who are interested in combining the study of biological systems but integrating quantitative methods, both theory as well as computation, feel free to reach out. This is a 3-year-old program, so these are pictures of 3 cohorts. We have 25 students in total. About half come from backgrounds in biology and half come from physics, mathematics, and engineering. And because I will be going through a number of slides, also this is more pedagogical material, trying to give you all background, I'll occasionally highlight some of the folks in my group who do the work. This is a relatively recent photo taken of our building at Georgia Tech. And if you're not familiar with the area, Georgia Tech is an engineering school. Historically has become a much more lively place to do science and basic science. Located in Atlanta, Georgia, with the southeast United States, right in the heart of the city. And this is a fairly typical shot. We have a mix of biologists, mathematicians, physicists, and bioinformaticians all working together on a common set of problems. So with those in mind, let me get into what I want to talk about today. And because these are staggered as parts of 4 lectures, in a few minutes I'll tell you what I'm actually going to do over the course of these 4 lectures, but I'd like them to connect. So today we're at the beginning. I'd like to get through some foundations of viral ecology and evolution, and bridging both principles and applications. So I want to tell you some of what I think are some of the important ingredients if you want to get involved in understanding quantitative modeling of viruses and the microbial host, but I also want to give you some of the payoff, what we are doing in practice to try to bring that to bear, both to understand environmental systems as well as increasingly therapeutic systems. So to begin, I'm going to show this picture, which seems like a strange place to start. Some of you who like to drink Negro Modelo may be familiar with the most interesting man in the world. That's this individual. He has all sorts of adventures. And the reason I'm showing you this is I want to contrast it with perhaps the most boring PowerPoint slide in the world, with just a bunch of numbers. It says experiment on the rows, replica on the columns. But I want to make the point that despite the fact that this is quite visually boring, I argue that it's perhaps the most important biological table in the history of biology, or at least modern biology, ever, which is a bit of an audacious claim. And before I explain what this is, has anyone or recognized has seen something like this before? Maybe the students rather than Paul Rainey in the back, who I'm sure has seen this before. But if a student, yes?
Is this coming from the group?
So Luria-Delbruck experiment. So that's right, let me explain what that is. I want to draw your attention to experiment number 5. And the numbers that you see in the different replicas are the number of colonies that are resistant to death by this virus that they've exposed and should be killing off all the bacteria. And you notice it was the same experiment. They did many, many replicas, and sometimes they got no resistant colonies, sometimes they found 1, and sometimes they found hundreds. And I don't know how many of you have done experiments before, but when you do many experiments, and sometimes the answer is 0, 1, or 500, or 200, that's not something you like to share with your PhD advisor, right? That just means it's all messed up. The brilliance here is that this variation was in fact the clue of some very fundamental biology that was taking place. And they designed this experiment, in fact, to ask, were the experiments repeatable? And the lack of repeatability was a hallmark of a particular kind of molecular mechanism distinguishing selection from mutation. So let me show you where I've taken this table from. It's, as was pointed out here, from the work of Luria and Delbruck. And you see at the top mutations of bacteria from virus sensitivity to virus resistance. And they did this many times. And again, the hallmark in each one of these experiments across multiple replicas was a finding of variability. And I should point out, I'm not just sort of making up the idea that this is an important table. This was one of the hallmark reasons that they, with also Hershey Chase, received the Nobel Prize in 1969. So let me explain the context. At the time, they wanted to understand, was mutation the change of an organism, ultimately its genotype, which revealed itself via phenotypes, independent of or dependent on selection, right? Did you have to interact with this either opportunity or threat to mutate, Or is that happening all the time in the background, and then it was revealed when the organism that was different was exposed to a different selective force? So let me give you two hypotheses in this pictorial form. Again, these are bacteria, and I'll be talking about bacteria in relationship to viruses over the course of my 4 lectures. And you can imagine starting with a single bacterium, which is enough to make a colony, right? These were originally called colony-forming units, but it turns out you just need one. And this bacteria starts to propagate. And the open circle bacterials will denote the fact that those are sensitive to this virus. Right? And this virus, if it infects this bacteria and it's sensitive, will kill the bacteria. You grow and grow and grow, put them on an agar plate, right? And then expose to viruses that should ostensibly be able to kill all of these bacteria. Now, if it turns out the mutations depend on selection, that dependency connotes the fact that with a very small probability, the virus will get inside, something will happen, and now all of a sudden this will have an immunity or a resistance to both that infection and future infections. And so that bacteria will survive the interaction and propagate, and on your plate you will see a colony of resistant bacteria. And here, this example, there were 3. So this is a notion of that the mutation and selection are intertwined, that mutation depends on selection. And you might think, if you know anything about biology, you might think, well, this is crazy. I'll say in a moment why not so crazy. And certainly this was not well understood at the time. In contrast, you can imagine now that mutation is independent of selection. That means in the proliferation of this one bacteria towards many, somewhere in this proliferation there will be already a resistant mutant that has arisen. And here we're assuming that there's not a significant cost to growth. And it will give birth to 2 daughter cells, and then to 4 and 8. And all of a sudden, instead of having just a few at the end, you may have a large subpopulation, all of whom are resistant. And when you spread them out on the plate, they will be at different locations. You'll expose them to viruses just as before, and all of a sudden you will get this jackpot effect, where you'll have a large number of resistant mutants. How large it is will depend on how far back in time this potentiating event happened. If it happened recently, we'll just have a small number of mutants. If it happened 4 generations ago, or 6, it'll be 2 to the 4, 16, or 2 to the 6, 64, etc. And the difference between these two, I should point out, is that when you do this experiment again and again, you have a small number, many trials, and I don't know if you reviewed binomial distributions or Poisson distributions last week, but that is you have a small number, a small probability, many trials, you get very repeatable outcomes. You're always going to get essentially the product of the very large number of trials, the number of bacteria you have, times the probability of mutation. Whereas in this case, you get a totally different distribution. I'll just give you an example. The data itself is, I think, the best example. This is a simulation that you could do in which you just do the same propagation. This is the best fit Poisson distribution, which is what we expect if mutations are dependent on selection. And the actual simulation, just like you saw the data, has this long tail where again and again you get these large number of mutations. So you have high variability is characteristic as opposed to low variability, which is why they ended up realizing through the actual lack of repeatability, the fact that when you look across this row, you see a lot of variation, was the hallmark of the fact that mutations were independent of selection. And so again, this is now what we understand mutation selection to be different. First we have mutations, which generates variation. Selection acts upon that variation. It is worth thinking through, given the rise, and this is not on the slide, and the advent of interest of CRISPR, which some of you may have heard about, which is an acquired heritable immune system, if you allow me to use those words for bacteria and archaea. If they had done this with Streptococcus thermophilus, it would have turned out a bit differently. You might have expected to see quite a different outcome. So the point that I'm trying to make, at least at these beginning slides, is that viruses, in addition to teaching you some hopefully basic bio and putting it in the context of viruses and bacteria, that viruses impose a strong selective pressure. Right? Viruses and microbes are lethal agents, and so therefore, host mutations that confer resistance are beneficial. Right? Those hosts that don't die when exposed to viruses are going to be around, and then they can begin to propagate. Let me just finish one more thing, and then I'll take the question. And so in a fundamental sense, viruses can induce host evolution, the change in the relative frequency of genotypes within a population. Right? So all of a sudden we started from a certain very low frequency of resistant mutants and all of a sudden after the exposure to viruses, we've totally shifted the population, and that can keep going on and on. And yes, I saw a question, Marcus.
So this mutation happens before the bacteria
Correct.
is exposed to the virus. So
Correct.
this is why you say this mutation is independent of selection.
Correct. So the idea is that you didn't have to, even if we had not I mean, this is the old adage about forests and trees, but if we had not added the viruses and we had done some very deep sequencing assay, we would have found, there really were mutants in that population, but it took selection to reveal the fact that they were there in this case, and then if we let them propagate, we could have shifted dramatically the population. But those mutations were there before you added the viruses, and in fact, a lot of the debate at the time, because the mathematics underlying, I'm not even showing you all the mathematical details of this Delbruck distribution, the mathematics were, in some sense, unreachable by the majority of people working in the field, so it took actually the work of Esther Lederberg, who developed the replica plating technique to show that the positions of where the mutations were also proved that these were independent selection and not somehow some pre-existing property. So it really was actually the visual proof that made people believe it. But to go back to your question, they were there. If you had sequenced them, you would have found the mutations even before you had the viruses, but at very, very low frequencies. Other questions? And like Luca said, although there's a question time reserved at the end, feel free and welcome questions during the lecture itself. Okay, so let me just get back into my train of thought, which is viruses impose a strong selection pressure, and therefore we have this host mutations that confer resistance are beneficial. Viruses are inducing host evolution, and this is well known from this Luria-Delbruck experiment. But what about the viruses? So the Luria-Delbruck experiment does a great job of revealing this independence, but the viruses are also out there doing things, and if all there was the Luria-Delbruck experiment, we would have just eliminated this particular kind of virus forever. Now we have this resistant bacteria, the viruses are done. So in a perhaps less well-cited and recognized paper, this is just by Luria, they went back and examined, in essence, the beginning of a coevolutionary dynamic between viruses and hosts. And just to get everyone oriented here, these squares represent the viruses, the phage. I'll use the word phage, bacteriophage, to denote viruses that exclusively infect bacteria. And the ovals are bacteria. The labels denote the type of bacteria or phage. And you'll notice that at the beginning there was this alpha type which could infect the beta type, and another type, the gamma, that could also infect the beta. And what happened was that you will see that these beta bacteria, there was an evolutionary event, a mutational event, and now different variations can no longer be infected by certain kinds of the viruses. So if you see a solid arrow, that denotes the fact that that particular virus can infect that particular bacteria. So if you see the absence of an arrow, that denotes there is a resistance event. And so one of the hallmarks here is that the host range of viruses can expand. Yes, there's a resistant mutant on the bacteria side, but they were able, by now trying to grow viruses on that resistant bacteria, to identify viruses that could infect that now previously resistant bacteria. And this was true both for the gamma and the alpha kind. But the other interesting thing that happened in this experiment is that when they did this in successive rounds, eventually they were able to identify a bacteria that was resistant to both kinds of the gammas and both kinds of the alphas. So all of a sudden, and they were not able to isolate a virus that could infect it. So you can imagine successive rounds, eventually we get this kind of bacteria, and they were not able, first of all, it's resistant to all four, there are no straight lines, and they were not able to identify a viral mutant of either the alpha or the gamma kind that could infect this kind. So this implies that this phage-resistant host strain emerges. This, although it was in the 1940s, right, 70 years ago, ended up becoming quite influential. The idea is that the accessibility of mutations for bacteria was supposed to be easier than that for the viruses. And so this became, in essence, dogma, and I'll give you one version of dogma from 40 years later. The coevolutionary potential of virulent phage, these viruses that infect bacteria, is less than that of the bacterial host. And if you know anything about evolutionary ecology, you've already heard the name Richlensky, Bruce Levin, so these are quite well-known folks who thought quite a lot about these problems. And there are many reasons why I think this was a tentative conclusion. Partially because it was very hard to find any evidence that these viruses of microbes were common in the environment. They were quite useful to understand molecular genetics and the foundations of molecular biology, but there wasn't much evidence they were actually very common in nature. And so something happened just a few years after this particular quote. And I'll show you right now what it is that was found. So in 1989, a group of microbiologists interested in environmental microbiology and virology began to look in a different way at natural systems. So this is electron micrograph taken from aquatic systems. They've looked both at lakes and offshore waters in 1989, and anything that has DNA inside it is stained in this sort of a dark color. And you'll notice also, importantly, the scale bar, 1 micron. So there are things that look like maybe they're microeukaryotes, prokaryotes, bacteria, and so on. What they noticed, though, were these circles that also seemed to contain DNA, but were circular and looked like they were about 50 nanometers in size. So they started to count how many circles that were there on one of these typical micrographs, and then doing the back calculation to say, well, how much water was in one of these particular fields of view. They made the estimation that they found 250 million virus particles or virus-like particles per milliliter in natural waters. That's a lot of viruses. And the other remarkable thing is that this was 1,000 to more than 10 million times higher than previous reports. Okay. So that definitely is going to change your view of how important or relevant viruses are going to be in natural systems if you've been off by factors of thousands, tens of thousands, or millions. And so this also raises the question, how could prior work been off by that many orders of magnitude? And part of the reason is that beforehand people were looking in a different way. So if you want to count viruses, like I explained a little bit of how they counted bacteria, you count bacteria by plating them and counting. Now there's something called this count fallacy, or a gap between what one counts and how many bacteria are there in a natural system. There are many bacteria we can't isolate. To measure viruses, you can do it in the same way. You give them food, which is some prey or host organism, you put those viruses down, and they make, for a physicist, sort of like antipar, a hole instead of a plaque, so they make the absence because they're killing off bacteria, and you count these numbers of plaques. The problem before is that they were often using things like Vibrio, which is a marine organism, but other organisms like E. coli, essentially trying to grow viruses on organisms for which they did not proliferate. And here they're looking in a culture-independent way, just looking for evidence. Now, it's not entirely direct because these are virus-like particles. There's some controversy there. But they were looking for evidence that there were viruses irrespective of what their host was. And so this is the beginning of what became a sort of a modern tilt. And if you look at the number of both citations, the activity interest in the field, it's at this point that a lot of interest gets generated. The other reason why there's been an increase in interest is about 10 years later, people began to realize that viruses were not just mortality agents, but may also have stimulatory effects on ecosystems, particularly marine ecosystems. And part of that reason is that when a virus kills a microbial host, it doesn't convert all of the carbon, nitrogen, phosphorus, etc., into more virus particles. In fact, it tends to use very little of the carbon. And then the rest is then released back into the environment. Now this becomes dissolved organic matter that can be taken up by other heterotrophs, in some ways stimulating the microbial loop and shunting away carbon from higher organisms. This became known as the viral shunt, and estimating the magnitude and the type of nutrients that are being redirected is still a quite important point in modern viral ecology research. So we have numbers, we have function, and then we also have the diversity question. So in 1989, when Berg et al. Looked and just counted these very large numbers, 250 million virus particles per milliliter, they didn't quite know who was there. They were just circles with DNA inside. And so it took adaptation of metagenomic approaches which had been used for cellular diversity analyses to analyze the diversity of viruses to begin to realize that most of this stuff was unknown to science. So this is a quote from one of the earliest studies of viral metagenomics by Maya Breitbart et al. Maya is now faculty at the University of South Florida has done quite a lot of work over the past 20 years, building on some of this early research in which 3/4 of all the sequences that they found had never been seen before. We couldn't necessarily characterize what they were. Now that situation has changed a little bit in the sense that in some cases we've seen it before, but it doesn't mean we know what it is or what it's doing or who it's infecting. Okay? So this is just to give you some sense of the background moving to the present of what viruses and microbes are doing, what they could be doing, and how much we still have to know. And before I get into the main features of the lecture, because again, I'm trying to use this time to set up all 4 lectures, not just today's. I also want to just step back too, because you may already have some preconceived ideas of what viruses are, and I want to make sure I direct our attention to the scale of problem that I'm going to be focusing on in my lectures. So what do we talk about when we talk about viruses? I would say that, hopefully you've talked about viruses, or maybe thought about viruses before, but you probably haven't been talking about phage or viruses of microbes. You may have been thinking about things like Ebola, Zika, influenza, other agents, whether it's mortality, morbidity, not necessarily good things that you don't want to get, right? And they're in the news because they infect humans. They can infect other species as well, but certainly they have direct effects on human health. So I want to make the point before I begin to think about and talk about viruses and microbes, that viruses infect organisms across the whole tree of life. Right? So there are examples of viruses of plants, of archaea, of insects, of microbes, right? Birds, mammals, etc. And I'll just do one last little anecdote. If you really get into these things, there's a great NPR piece, it's in English, but it goes slow, it's quite nice, on viruses of insects. You may be familiar with the book, I don't know how well it's been translated, The Very Hungry Caterpillar. You may have been read to it, or you may have read it to a child, or a niece or a nephew, etc. So it's a great story about this caterpillar that eats junk food all week long, and on Saturday sits on a leaf, eats the leaf, feels better, and then begins this process of becoming a butterfly. So there's a director's cut version of this story in which the leaf that it landed on and began to chew had some baculoviruses on it. And these baculoviruses are interesting because they infect caterpillars, other insects, and they begin to spread inside the caterpillar, redirecting its physiology. It begins to climb higher onto the tree and ends up on a leaf where the proliferation of viruses becomes so intense that it ends up turning into essentially virus goo, and then the baculoviruses rain down and fall from one leaf to another leaf where its life cycle can begin. So if you want to learn more about baculoviruses, it's quite an interesting story. So what I'm going to try to do over these next lectures is introduce you to a planet of viruses from the perspective of ecology evolution. And the front matter piece here, this is a book by Carl Zimmer, is that of a cyanophage. So cyanophage are viruses that infect ubiquitous marine bacterium that sit at the very base of the ocean food web. And these too get infected by viruses. So what I'm going to do in the lectures today, Wednesday, Friday, and Saturday. What I'll try to do today and Wednesday is to focus on principles underlying virus microbe dynamics, trying to link 2 different concepts, ecology and evolution. Okay? And I'll do that in 2 parts. Today I'm going to focus on relatively simple ecoevolutionary dynamics, and then on Wednesday build up in complexity and think about dynamics of complex communities involving multiple kinds of viruses and microbes interacting together. And for the most part today and Wednesday I'll be emphasizing the lethal lytic component of viruses. That is, like Lorraine Delbruck, focusing on viruses that infect bacteria, kill them, reproduce, in other words, make progeny, and then go on to find a new host, and the consequence that it has both on the ecology side and the evolutionary side. But on Friday, I'll switch gears and try to broaden these sets of lectures and think about the contrast between lysis and latency. Viruses that integrate into host cells and begin to replicate with the host cell rather than killing it, and the consequences that has both for ecology, evolution, but also life history traits. What are the optimal ways that viruses and other parasites should make use of their hosts? So that'll be on Friday, from lysis to latency. And then on Saturday, and I realize it's the very last lecture of this course. I'm already anticipating all of you being exhausted. So I will try to have something modular, self-contained, that emphasizes the ways in which we can bring some of these theoretical ideas into application directly in terms of therapeutics. And I'll try to convince you should stay for that one, because I'll be talking about ways in which phage specifically are being used as alternatives to antibiotics in the wake of this advent and increasingly number and crisis within antimicrobial resistant pathogens. So I'll try to explain both from the theory side, which hopefully these other lectures will prepare you for, how that can be put into this new context, into whether it's a human context or other mouse models. So throughout, what I'm going to try to do also is theory and modeling motivated by fundamental eco-evolutionary challenges and also with some of these real-world applications. So I think that's a long enough semi-intro, both giving some background, but let me get into it. Are there any questions before I move on? Yep.
Can you just define what is a virus?
Yes. So would you like all viruses or just phage? So a virus, let's first talk about parasites, and I'm going to give the life cycle in a moment. So parasites are organisms that rely on a host for reproduction. Viruses are obligate intracellular parasites, meaning they must, in order to reproduce, get inside a host organism in order to replicate. Most of them, when you look at virus definitions, they include some sort of packaging of DNA or RNA genetic material inside a capsid. The capsid is usually protein containing, but it also can have lipids and other materials. So for the purposes of these lectures, we should think about viruses as obligate intracellular parasites. They must get inside a host to reproduce, and nearly all the viruses I'll be talking about obligately infect bacterial cells, and I'll focus largely on double-stranded DNA phage, which are enveloped when they're in their particle phase inside a protein capsid.
Can a virus die?
Can a virus die? So now you're asking me, in other words, are viruses alive? Because for me to say, and they can decay and they can lose infectivity, and that will be part of, so I'll answer by saying yes, viruses can decay and lose infectivity. If you make me tell you that they can die, then you've asked me about life, and then I'm in trouble, and then we can't move forward. But yes, but they can decay. And they absolutely, in fact, many of them aren't even infectious at the beginning, but that's a different story if we want to talk about that. Okay, so what am I going to do in today's lecture? I'm going to try to go through a few concepts. How does viral infection change microbial population dynamics? And in turn, how does co-evolutionary change alter these dynamics? So I'm going to first start with ecology and then get towards evolution. And then I'll build up, depending on the pace today, into Wednesday most likely in this question of dynamics in complex communities. Okay, so part one, how does viral infection change microbial population dynamics? And to do that, I'm going to also still go back in time, but purposefully. About 80 years ago, you probably heard last week about Walkerville Terror. I think that was on the curriculum. Is that right? Please nod. So you know that Vito Volterra was convinced by his son-in-law, Umberto d'Ancona, to look at fluctuations in the Adriatic fisheries. And the question there was, were these oscillations, the amount of catch, a byproduct of something intrinsic to the system, right, or something extrinsic? Were there oscillations in some seasonality or other conditions that were external to the system? And his proposal, which is written out here, is that he considered a case, this is Volterra in 1926, two associated species of which one, finding sufficient food, would multiply indefinitely if it was left to itself, while the other would perish for lack of nourishment if left alone, but the second feeds upon the first, and so the two species can coexist together. So in essence, they're very basic foundations of these predator-prey dynamics. And in a modern way, we would write, as I think you learned about last week, a coupled set of ordinary differential equations that happen to be nonlinear, where n are the prey abundance, p are predators, the dot denotes, I'll use dots or d by dt's to denote derivatives with respect to time. And here we have the left with a lot of food would grow unchecked. Here we have feeding on another, leading to more predators. And then we have, if by itself, it would die. All right, so we have all the ingredients of what Vito Volterra's model had. And the consequence, if you look at time and populations, you will see these oscillations, where prey increase in abundance, and then predators increase because there are many predators around, then prey decrease because there's an absence of prey, then predators decrease, prey recover, and the cycle returns again. If we look at this in a phase plane, here we plot prey and predators, and the axis of time is implicit, you'll notice it makes these counterclockwise cycles. And that, again, seems fine. We get these Walkerville Terror-like oscillations, but it's actually not so fine because this system, as originally developed by Walkerville Terror, was a conservative system, which for physicists is good news. That means there's some symmetry in the system and we can probably work with that. But here, it implies that the initial conditions last forever. If we start with a different combination of predator and prey, that's permanently inscribed in the system. The system has infinite memory of these initial conditions. And that's really part of the fallacy of this particular choice of the right-hand side set of equations. So later models began to fix that. First of all, you could just have some handling time. If it just takes some time for the predators to handle the prey, you end up getting limit cycles, and this is data from the Lynx hair system, where lynx eat hair. And again, the point you should take away is that we have time, we have prey, predator. There's an offset, there are oscillations, and the implication here is that these oscillations are driven by feedback between predator and prey abundance. That is what I think you covered to some extent last week in an abbreviated sense. Okay, so it turns out that this work of Walkerville Terror was strongly influential and remains so in understanding virus micropopulation dynamics. So this is work by Bruce Levin in 1977, in which he, with collaborators, in one of the earliest papers, I think if not, maybe perhaps the second earliest paper, conceived of microbial systems as microscopic versions of these predator-prey dynamics, in which instead of observing over a 50-year time period, you could manipulate over a period of a few days or weeks. And the idea here is that there was one resource, for example, a carbon substrate, one prey, which was the bacterial host, and one predator, which is the virus. And they intentionally use the word predator, thinking about both Walkerville Terror-like dynamics, but also the notion of a predation, of a killing involved in the replication of the virus. Okay, so the idea here is that the prey are the bacteria, the viruses are the predators, and to move even further back in time, this is work by Alan Campbell, who described viruses as a simple predator. Although there are different variations of the model, this is the one that I want to start us with. Again, you'll see now the connection between this work and the work of Walkerville Terror. This is a dynamic model in which we have three components. Resources, that resources which the prey is eating, consuming to reproduce, the prey, the bacteria, and the predator, the virus. This is an environment where you notice here that there's resource being flown into the system with some density R0. There's, R0, I shouldn't use R0 because I'll use that later, R0. Omega is leading to the decay or loss of all three kinds, and that's because here, these initial models were done in chemostats, I believe they were asked about before. I'll describe them in a little bit more detail in a moment. And in the chemostat, only resources are being put in at a rate omega, but everything in the chemostat is leaving at a rate omega. So the resources, the prey, and the predator are all leaving at a rate omega from the system. Resources are being taken up, converted into new bacteria. These are being infected. The virus particles are removed temporarily and then released back with a burst size beta. And here in the simulation, you can see that resources and prey are denoted by blue and green respectively. And when you add viruses, the resources increase, the bacteria drop down, and there are oscillations. The reason is that now instead of the resource level being controlled by the bacteria being controlled by the viruses, and therefore releasing the pressure. So we go from a bottom-up to a top-down situation, where now the viruses are controlling the density of the bacteria, so they no longer draw the resources down as low as they had done before. I can move this, although this is a three-dimensional system, I'm going to ignore the resources for a moment, and just look at the bacteria and the virus dynamics, again plotted in the phase plane, and you see these characteristic counterclockwise cycles, right? Where, again, we have not that many viruses leading to the reproduction of bacteria. As there are more bacteria, we have a lot of viruses. We drop down in bacteria, and with very few bacteria, the viruses start to decay fast, and then the cycle repeats. Now, there are many issues with this model. I'm introducing it because it's simple to present and understand, But now let me go and hopefully answer a little bit more about this question of what is a virus, what are the life cycle of these viruses. And you'll begin to see there should be some modifications. Here we want to ask the question, are these kind of conclusions robust as we start to inject more of the microscopic mechanisms by which viruses exploit their microbial hosts? Okay. So what is the life of a bacterial virus or a phage? Again, I'm going to use the word bacteriophage or phage from phagos, meaning to devour, but usually I'll just say phage. We can think of this as about a 50 nanometer passively diffusing particle that gets in contact with the microbial cell, injects its genetic material into the cell, and then that genetic material redirects the host cell to start reproducing the viral genome, as well as proteins, RNA that are required for the self-assembly of the capsid, and also the spooling in some sense, or the packaging of the genetic material of the virus inside that virus particle. And then through a timed process, they release either holins or lysins, which make a small hole in the inner membrane and can also make an incision or cut bonds in the cell wall. And then these virus particles are released back out into the environment, and the cycle repeats. Okay? There's a time it takes to get from here to here. This is called the latent period. Yeah?
What happens to the original virus?
The original virus, and that's going to be a theme, the original virus and this virus particle, its genetic material is here. A virus is not a particle. Right? The viral genome is the unit under selection. Right? So the particle by itself, and that goes back to obligate intracellular parasites, that virus particle without its genetic material inside is no longer has an evolutionary future. The genetic material inside the host, this infected cell, then is going to be the route potentially to reproduction. Other questions about this? Yep.
Can the cell functions be debilitated?
Start over.
Okay.
Yeah.
You have the whole cycle. However, can the virus be... He makes like 4 steps, but he cannot reproduce itself. However, it kills the cell anyway. Can it happen?
Kills the cell. I think I understand the question. Can the virus kill a cell without reproducing? Is that what you're asking? Okay. So the answer is yes. In fact, some cells may even kill themselves before the virus kills it. Right? So there are these apoptotic pathways and various reasons why, I'm not going to say it's beneficial, but we see sometimes that the apoptotic pathways may initiate so the virus never completes its life cycle. There also can be killing from without. So the virus has to inject genetic material. In some separate work I'm not going to get into today, we looked at how big holes can be within a cell wall before the cell loses integrity, so you can get lysis from without. There's too many viruses outside and the cell can die. And also these are stochastic processes, so it's quite possible, though it wouldn't favor that particular strain or genotype, for this cell to die before it reproduces. Over the long span, that would not be an effective way to reproduce. You go inside, it sounds like spite, and there can be evolution of spite, but at some point there has to be a successful infectious particle. So it can happen, but if it happens too often, you're not going to have viable progeny. Okay. So again, to point out that these models were done in the context of a particular environment. In the environment that, for the most part, these simple both experiments and models were conducted in are this notion of a chemostat. So now let me just describe what a chemostat is, in which we have media reservoir, where media is being put in, usually in these cases, let's say a simple carbon substrate, a single carbon substrate, and then everything is taken out so that you can then measure both in the effluent or even what's coming out of your sample probe, the current status in terms of the density of resources, microbes, bacteria, and viruses. Okay? But I had shown you before that there's a gap here in time between when viruses get in and when they get out. So the original model has these viruses acting like predators as if they're eating the prey and instantly turning that into new biomass. So this is a variant of these same models with an explicitly infected class. And here we have, I've gotten rid of the resource layer, in which there are susceptible cells, infected cells, and viruses, virus particles. The susceptible cells can reproduce. Here's a logistic-like growth model. They get infected, and all three are being washed out of the system at a rate omega. These infected cells then increase the density of infected cells in the population. And then there is some time, characteristically 1 over eta, over which those infected cells will lyse, leading to the generation of beta new viruses. Right, so now I've explicitly put in essence, an exponential distribution of decay times of latent periods. And again, here the viruses go in, so they're losing virus particles and regenerating virus particles because there's an infected class that then at some point later, exponentially distributed, will produce a burst of beta viruses. Well, there we go. Let me just go back one. And so what the outcome here is, here I'm looking at total host versus viruses. Then now instead of, let me just finish this one thought and we'll get your question. Instead of seeing a spiraling in, we get limit cycles. So for those aficionados, there's a hot bifurcation in the system, and so there's a robust parameter regime where you see these limit cycles. The point I want to make here is that these are oscillations remain counterclockwise. So they are the characteristic Volterra-like dynamics, but now instead of being neutrally stable orbits, you get this attracting limit cycle. Can I finish the next half slide, or you had a question there? Right, so in this case, I have put all of the mortality, assuming that the washout rate is faster than whatever the characteristic death rate is, either of infected cells or host cells. So host cells don't last forever, but so the washout rate is going to be the dominant feature. I could change the model and have omega I plus some other small value, and it really won't change things that much. It would be a rescaling. The same thing applies here. When we work in natural systems, one of the big questions we have are how long virus particles last and how long they're infectious. Here, the decay of virus particles on the bench is usually quite long and longer than the characteristic time scale of these, of the turnover of the media, which is really setting the decay rate here. But you can add those features too. It doesn't really change things. Yes? So here would be a resource-limited carrying capacity. I haven't had the resource layer, but it's not like these chemostats are going to be filled up with bacteria. They're going to run out of resources well before they'd run out of physical space. So here I don't have the resource layer because then I have a four-dimensional system. You can actually reduce it to this sort of model and you get this effective carrying capacity that has to do with the uptake efficiency, the resource supply, etc. Okay, so this is one microscopic model. The problem with this model is, if you know anything about viruses, then you'll realize that viruses, after they get in, they don't immediately come out. And when I have this minus eta term, I have an exponential distribution, which means the most likely time for the burst is right now, right? So another way to do that is to put in an explicit delay period. And this is often done by taking these same three component models, susceptible cells, infected cells, and viruses, but instead of having a constant probability per unit time of bursting, here what I have is an explicit delay differential equation in which new infected particles, new infected cells are produced at a rate phi nv, and then new bursts happen based on how many new infections occurred tau ago, conditioning upon the fact that only a fraction of them survived tau before the burst happens, because some fraction of them being washed out, right? So that's all that is a mathematical difference. You can also play these games by, instead of just having one infected class, you can have two, and you can have a poor man's version of some sort of modal distribution with a peak in the middle. So this is a delta function distribution, the other one is a exponential distribution. The point, though, is that despite some quantitative differences, you once again get these Volterra-like dynamics in which we have these characteristic counterclockwise cycles. Okay? So again, tau is the latent period. So these are continuous differential equations, but tau would be, and it could be 20 minutes to 40 minutes to half a day, depending on the strain. Right? So often case for these viruses of E. coli that are growing in rich media, the latent period could be anywhere from 20 minutes to 1 hour, so it could be quite fast. For slower growing host strains, it's slower. Okay? Yes?
During the latent period, we assume the virus is still on one individual, even though like it might be reproducing inside the bacteria.
So this I denotes an infected cell. So we have one infected cell, and that infected cell is going to produce beta viruses, new virus particles.
Okay.
Right. So the death of that host cell leads to beta times the density of host cells per unit time times beta is the density of virus particles produced per unit time.
So the delay in this differential equation is represent the probability of cells being alive but not explicitly on the derivative.
Right. So here, I mean, to simulate or solve delay differential equations is its own. To solve or to simulate them takes a little bit more work. But let me explain again what I mean by the notation. So here the left-hand side still denotes the density of cells, whether susceptible, infected, or viruses at a given moment in time. So here we have an increase in infected cells right now based on new infections. But we're going to lose some of those infected cells because some of them are going to burst. How many? We have to go back in time, tau, so that subscript tau denotes, you could think of it as n of t minus tau. Okay? So n tau denotes n of t minus tau, v tau denotes v of t minus tau. We go backwards, time tau before, and say, how many new infected cells were produced at that time? Those should be ready to burst right now. The problem is, not all of them made it, because it's a chemostat with a washout rate omega. So because of that, you can see we have to ask, well, what fraction of those infected cells tau ago are still in the chemostat? And the answer is e to the minus omega tau. Right? Because the longer we wait or the faster we turn up the dilution rate, then a decreasing fraction of them are still left. So sometimes you'll see this without this term, and that's bad. So you need both to go backwards in time and then ask how many of them are still there in the experiment. Okay? So just to get us to a point where we're all on the same page, I went from Walkerville Terror-like dynamics to some of the very basic mechanisms of virus microbe interactions and added more and more features, all of which are maintaining this characteristic feature of these Walkerville Terror-like dynamics, which seems to be robust, in which we have a small number of viruses which will lead to the proliferation of bacteria. As there are more bacteria, we get more viruses, which drive down bacteria, which drive down viruses, and the cycle repeats. Okay. Now, again, it's going to get more exciting, but I still need to get all this stuff through. And the other thing I wanted to do was a bit of a check, because before I'm out of my time today, I want to show some data and kind of lead it to the next lecture. But there can be a false sense of security here that if I have a virus that infects a bacteria, it's inevitable that it's going to proliferate. So I want to ask this question of what viral life history traits enable the invasion in the first place. How does this virus even get established? Is it inevitable? If I have a virus that infects the host, is it just going to happen? Right? Are we going to have the proliferation? So that's what I want to do, and I was told I should consider using the board, so I will consider using the board. And I think that, I actually think it's okay. Which is it easier to see with or without that light? It's better like this. Okay. So what I want to do is to think through this system in which we have these susceptible cells, infected cells, and viruses, in which we're making transitions. This is a compartmental model of the kind I think that you're talking about last week, where this transition depends on viral infections, and we have loss out of each one of these terms, and we have some of kind of reproduction event. Right? So here we have, at least in this model, we have R, I'm using omega. I'm sure in my notes I probably used a different, of course I did, I used a different variable, that doesn't matter. So we have, I'm going to write these as D, make these different, then explicitly now separate out the weights of the decay of the viruses from the decay of those. You can think of omega plus some other thing if you'd like. Okay. and here we're going to have some beta at a rate, so this is our rate, eta. Okay, so here's our setup, and we want to know what's going to happen when we add viruses into an environment in which they're not there. And to do that, we have to have our model, right? We have to have our three components, which it's nice there's a lot of board share. We'll be able to use these later. Okay, so we have these set of differential equations, and we're going to try to ask something about whether or not viruses can invade. And what we're trying to do in essence is ask the question, if we start, I'm not going to worry about the y-axis, I'll get rid of it in a second, and we start with an environment for which there are k density of microbes, and we add a small number of viruses. The question is, will we go somehow into this interior of the phase space, or will the viruses proliferate, or will you just have a microbe and media flask, you've added some viruses, your experiment fails, it doesn't proliferate. What does it depend on? We already know that if you didn't have viruses in the first place, this is seemingly a stable fixed point, and from an experimental point of view, all that means is that yes, you can grow bacteria in a flask, right? And then if you dilute them and do it again, you'll do it again and again and again. If you do it a lot of times, you'll actually do an evolution experiment, but that's a different story. We'll get to that. Okay, so in this particular case, we want to ask what happens in this three-dimensional system. I'm going to make one trick. It turns out this trick works, but it makes it a lot easier for me to do this part, which is I'm going to ignore the density of infected cells. I'm going to assume that this rate of lysing is so fast that this is a fast variable, and I'm going to set it to 0. And at that point, if I set I dot to 0, I find that the I, which I'll call as a kind of a quasi-static equilibrium, will equal phi and V over eta plus D, which just says that the amount of new infections balances the loss. I could do the full three-dimensional system, but it would not be as fun or as intuitive. So you just take my word for it that we are going to assume that the infected cells are sort of tracking the rate of new infection as we go. And the advantage of that is that we can rewrite this system now in terms of a two-dimensional system. We're really going to be asking this sort of question. I could have started there, but it's nice to see where it comes from. So we have Rn 1 minus n, and I have to do something there. I'll ignore that later. Minus phi V dn equals, now you see we can replace this. Okay. So we have this two-dimensional system, and we'd like to know, will the viruses invade or not? What you can begin to see here is that in the viral dynamics, we have V dot is a product of the new infections minus the loss of virus particles because they're getting inside cells, plus this other decay term, which is also dropped. And we could write it in the following way. Where we've pulled the V out of the equation, and you see this is almost like an effective growth rate, where we have increases minus the losses, right? And you can already begin to see that if this difference is positive, then we should invade. Okay. So we're thinking about viruses as having a host, and therefore they should proliferate on the host. It's true that one cell might die, but that doesn't mean necessarily that there's enough reproduction for the viruses to proliferate. So what I'll do here is now begin to rewrite things in a slightly different way. You can see I could also write this, the viral growth rate, as beta wiggle phi n minus m, where I've just redefined, for a particular reason, this as okay? And it's going to look strange, but there's a reason why I want to do it. So now it really looks like as if we had gotten rid of that infected class. We just have contacts with an effective burst size minus the mortality. Okay. Now there are two ways to go about this thing, and this is fine. If this is greater than 0, this will invade. But there's a particular condition we care about. The condition where we have a uninfected or initially totally susceptible population. Right? So now you begin to see that it depends both on the life history traits, but also the context, not just of the virus, but the context of the bacteria. Okay? Now, there are ways for me to do this at depth, but because it's almost 3:30, I just want to point out that if you don't have the intuition behind this system, it's not a bad idea to take this two-dimensional model, linearize it, find the eigenvalues, and then find the conditions at which the linearized version of this system has an unstable, in other words, positive eigenvalue. And I think you went over that, how to do it, and I'm happy, on the break, not so happy, no, I'm happy, I can do that, but I'm not sure it gives as much intuition once you know that procedure as what I'll try to do now. Okay? So here we have something that seems okay. It's at this population scale, but I want to now move to the individual scale and show why there's a relationship between this growth rate derivation, which seems a little opaque, it seems algebraic, and something quite intuitive about what really drives the host. Did that go out or no? No. Good. Okay. So let me just do this last bit, and then I'll try to provide some intuition. Okay. What this is saying is that eta over eta plus d minus 1 phi k must be greater than m. If that's the case, these viruses can proliferate. So viruses will proliferate, or they will grow in their population if this condition holds. Right? I can write this also as beta eta over eta plus d must be greater than 1 plus m over phi k. This is not very complicated algebra. I want to do the steps to show you how I get to this point. In other words, beta eta over eta plus d is greater than phi k plus m over phi k. Still very boring, but it's about to get exciting. Everyone following? Okay, good. Which in other words means the following. Okay, same condition. I want to give an interpretation behind what this condition means. And I want to think about a single bacterial virus that's out in the environment, and there's a susceptible cell, many of them, and this virus at some rate m will decay, maybe because it just gets diluted out of the chemostat, or it loses its infectivity, or a combination of both factors. But at a rate phi k will infect a particular cell. Now there's a viral genome inside of it. This infected cell also has a bipartite path or split path in some sense, which is that it may decay or die, washed out, or suffer some other form of death at a rate d, and it will have a burst at a rate eta, in which case we will have 1, 2, beta new viruses. And I claim that this condition is asking whether or not, once you go through this process, if you start with one virus, on average, do you end up with more than one virus particle? If that's true, that'll be true for the progeny, and then you'll get exponential proliferation. So what are the meanings of each one of these terms? This term here, if I have two rates, the probability of one process happening is just the ratio of that rate divided by the sum of the rates. So phi k over phi k plus m is the probability that the virus particle infects a host cell before it decays. Once it's infected, this eta over eta plus d is the probability that the infected cell bursts before it decays, multiplied by the average burst size. Turns out that this is what's called the basic reproduction number. It's also been called in the phage literature the phage reproduction number. We're trying to unify that, and you'll get to hear how to unify all this on Friday. The idea is that this is now an individual view. It's measuring fitness at the scale of an individual virus particle and asking a little bit like the Wright versus Fisher way of continuous versus generations, is saying in one generation, for an average virus, does it produce more than one progeny over its lifetime on average? And if this is the case, then in the context of these nonlinear differential equations, you'll end up getting more and more viruses, and you'll get to a new equilibrium. So that gives me, can I indulge and just go forward? There's one more thought. Can I get just a little bit further? So we have this notion of this dimensionless parameter, right? It has no units. If it's greater than 1, it's going to lead to proliferation. And we're going to go to some interior, which I claim for various reasons will be smaller than K, because it's killing off these cells. So the host density is being driven down. And then you could ask yourself, you can see from the model, all the way on that side, in the middle panel, which one do I want you to look at? Ooh, yes, the V dot equation, that n star should be equal to m over tilde beta phi. It should be drawn down to a new equilibrium. I don't want to even tell you what the V star is. It doesn't really matter for the person I want to say. So we're going to have a new density in the system. So I claim that we can actually use this basic reproduction number to understand why this must be, not just because the algebra tells us so, but why this must be the new density, why it's being drawn down to this level. And my rationale is that at equilibrium, the viruses, instead of making more than one virus for every virus particle, they better just make one, just replacement. So I can ask, if I have some effective reproduction number, which is now equal to beta times eta over eta plus d times phi n star over phi n star plus m, I claim that at equilibrium this should equal 1. And it turns out I'll spare you the algebra, but that's precisely what happens. So we go from a situation in which we have traits, both of the virus and the bacteria, that allow for the proliferation of this population. And the condition is one in which, if we think from the individual view, if that's more intuitive to you, you take a single virus particle, is it going to, on average, infect enough cells, survive the infection, lead to new virus particles, that leads to more virus particles than you had at the beginning? If that's so, that process will lead to the initially exponential increase in viruses and decrease in bacteria, and eventually a new equilibrium in which those processes are now in balance. Right? And this also allows me to telescope a little forward, which is that you should notice here that the way that these viruses are reproducing is strictly horizontal. They have to leave the cell, find a new cell, new host, in order to reproduce. On Friday, there's a couple themes I'm developing, but one of them is on Friday, I'll ask the question, what happens if they were to stick with that cell? what is their reproduction number then? Do they even reproduce if they don't kill the cell and if they don't make viral progeny? So I think that's where I wanted to leave this part. And I only have 2 more slides left. So that was the checkpoint. Ended up lasting a while. And where did my clicker go? There we go. So I just want to make sure that I don't finish, because there's a lot of data. We work very tightly with experimentalists, and I haven't really shown much data today. This is math models of evolution, but I feel there should be data involved. So I just want to point out that these analyses of virus microbial systems have been looked at, as I said, from the late 70s. This is one example from Brendan Bohannon and Rich Lemsky, in which they observed Walkerville Terror-like cycles between T4 and E. coli B. And again, I just want to point out here, these are oscillations of almost 2 or more orders of magnitude in population density with a chemostat-like constant environment, where all these oscillations, for the most part, at least at this stage, are being driven by the feedback between viruses and their microbial hosts. Okay? And you should also notice there's a shift here in which there's a peak, and to the extent to which there's measurement resolution available, the virus peak seems to follow that, or coincide or slightly follow that of the prey or bacterial peak. I also want to point out this is 200 hours of an experiment, right? So we're talking about 8 days or so is about that kind of length involved. Try to remember the slide because I will return to it on Wednesday. I know we're interleaving here. So the summary of this first part, the take home of what I wanted to get done today, is that these original virus host models presuppose a very simple interaction, which is contact-based encounters in which, like Walkerville Terror-like dynamics, the viral predator utilizes its host to reproduce. I've shown you that these Walkerville Terror-like dynamics are robust to inclusion of additional microscopic mechanisms, and also that the invasion and persistence depends not just on the fact that the virus can infect the cell, but on life history traits and external components like host cell availability, right? Because if you try to get out, even if you're very efficient at killing the cell, if there are no cells left around to reproduce on, there won't be sufficient cell densities for proliferation to be possible. There's a big however, and that'll be the connection to Wednesday, which is that evolution can rapidly change the number and relative abundance of virus and host strains. And so a lot of the things I've just described get more complicated and interesting once you begin to think not just about ecology, which is what I've talked about today, but also evolution. So I think that's where I want to wrap up. And I'll just point out that when I resume on Wednesday, I will begin to answer this question, which is how does evolutionary and co-evolutionary change alter viral host population dynamics? So happy to take a little break, and I think we'll stop there and come back after break with questions. Okay, thanks. So we have 10 minutes. We'll come back at 45. It looks like I went a few minutes over. 45 by that clock or so.
The last term of this question is just because the last. Oh, I understand.
Yeah, sorry. Sorry.
Thank you.
That was very nice. Now, I have some basic questions. What's the lifetime, for example?
Okay, so let's start the discussion session. Okay, so any questions or comments?
It was all perfectly clear, so you can go more coffee.
Why did you use the tau in M and V, and you are related to the delayed population?
So the question is, I'll just repeat the question if I don't get it right. You can ask it again, which is, or do you want to try again?
When you use the N tau and V tau, you are relating to the delayed populations, right?
Yeah, and it's just a little bit of a shorthand. Whenever I do this, I mean that. So, and part of it you can think about the release of viruses right now through a lytic event depends upon events that happened in the past. And in the first model, there's no memory because I have a rate eta, which if you think about what's called a generation interval distribution, or the probability that the time of infection lasts a certain length, if I have a fixed rate, then I'll have, let me be over here. If I have a fixed rate, then I'll have this shape, e to the minus eta t, right? Whose average time, 1 over eta, I can define as tau. So I could use the same average time, but in the second model, I've basically assumed it's a delta function. The reality is that it's probably something more like this. And so if I want to play with these models, I would have to choose something intermediate between these two extremes. And the simplest way to do that in practice is I could have an E class and then an I class, so an eclipse phase and then let's say an active infected phase. If you've ever done any epidemiological work, you can have exposed or asymptomatic or multiple classes. When you add these classes, you're essentially playing with the shape of the time, which is called the generation interval distribution. You could do the same thing in this model. I've taken two extremal cases here.
Who had a question?
Can you go back to the last slide, please? You were talking about. Oh, you don't need to. You were talking about the coevolution effect, and I know that's going to be a class only of that. But for the experiment, you showed that there were the delay in the predator, the behavior just like a predator-prey dynamics. Like it lasted something like 200 hours. Isn't this enough time to evolution matter?
So that's why I have a Wednesday lecture. Because this experiment shows something very interesting, but I can't give it all away today. But certainly you're right. Let's go back to Lurie and Delbruck. In Lurie and Delbruck, let's say in an overnight, you've totally killed off the susceptible population. Now you have a few resistant individuals. If you allow them to then regrow, remember, exponentials are very fast. They take about log of the population size multiplied by the division period, and that's how long you have to think that you can go from 1 up to back to the total number again, right? So here, part of that is going to be, there can be trade-offs with respect to different kinds of mutational events. So let's be clear here that in the background, there's a lot of variants that are sitting in this background. The question is, do we start to see those variants at high enough abundances that they've changed the dynamics? So the answer that we give at this stage is that soon after I've cut off the time series, something else is going to happen. And I don't have to ask the question what it is. It's going to have to do with evolution, right? So that's going to be on Wednesday, which I'll explain. But you're right. So these things are there. And now we've been doing some of these experiments, and once you go back in deep sequence, you find out, wow, there's a lot of interesting stuff in the background. But this is just based on the plate morphologies and colony counts. So this is limited in some sense by the technology at the time. I think we're going to end up probably revising a lot of what we thought about the pace of coevolution evolution as we start to see more of the variants that are there. And even rising to relatively high abundance is just not enough necessarily for us to see differences at this population scale. But I will, if I don't, I'm going to answer your question fully on Wednesday. Absolutely. I think we have one right here. It's right behind you. And we have 2 mics, so if someone else wants to send it around. Go ahead. Yeah.
When the R0 is bigger than 1, can I say that the bacteria will no longer will have a time that goes extinct, or?
Okay, so what does it mean to have R naught greater than 1? This is from the context of the viruses. So the R naught greater than 1 means that the viruses can invade, but they don't necessarily have to, because I'll go back to Luca Politi's argument about branching processes. So if I take this individual perspective, you can already see that with some probability m over phi k plus m, that first virus particle doesn't even infect, right? And then even if it infects, then with a probability d over d plus eta, it doesn't produce. So those still have 0. So the point is that when R naught is greater than 1, we're talking about an average case. If we were to run through each individual, we'd find that on average they would proliferate, right? But then if we have a birth rate and a death rate, then the chance of one happening for a branching process is 1 minus the death rate over the birth rate, says that it actually proliferates in the large population limit. So let me answer your question now in a succinct way, which is R naught greater than 1 means the virus is spread, but it doesn't mean the bacteria go extinct. Very important. And that's actually going to, again, telescope to Saturday, because when a virus invades, but it's an obligate intracellular parasite, it doesn't necessarily eliminate its host, because as it becomes more effective at eliminating the host, the densities go down and its effectivity also goes down. So there's a self-limiting nature there. And that's why that is the value of the equilibrium and not 0. Right? So that's a very important point. Yep.
Sorry, can you repeat why or under which conditions the dynamics of E can be considered like a slow dynamics? I missed the explanation.
Okay, so I'll answer more fully, which is that for the purposes of me showing you a 2-dimensional system for which then, if you had wanted to, I had in my notes actually me going through the whole operation, and I realized, oh, that's going to take a lot of time. The linearization of the system, you can linearize it in 3D, so the NIV system, you'll get this same threshold parameter. It's the same. Okay? So in general, so physicists sometimes like to play with these adiabatic approximations, where we assume one variable is slow and the others go fast. And even when they're not infinitely so, it tends to be a good approximation, certain limits. So here, it would be relevant when the latent periods are relatively short or fast compared to the change of the rest of the population dynamics. Now, it turns out even when that's not a perfect approximation, it still works pretty well. But with respect to technical, if I had gone through the full 3D system, I still would have found this was the threshold parameter. But then I would have had more information about the eigenvectors of the system, the directions upon which the I was changing. So we haven't led you astray by putting this answer out. That's still going to be the answer even in the full 3D system. Yep.
Professor, last week we, in my group, worked with a system which is an entomoparasitic nematode, meaning that it has to kill its host in order to reproduce. So it's very similar. And yesterday I was asked, and I unsuccessfully responded this question, which is, how do you think your results or the phenomena would change if you account for the process outside of the host? You know, that viral particle, that viral genome, I've never seen it considered in models, but how would dynamics change, if at all? Okay, so I'll try to answer your question. I'm sorry you had an unsuccessful attempt at answering your question the other day, but now maybe you'll get a successful attempt in the future. In this model, you'll notice this is the extracellular phase,
And you can think of this as the intracellular phase. And in fact, you can think of both of them together as the infected subsystem of the complete system. And in fact, when you want to do all this, and I will tell you a little bit more on Friday, when you want to analyze these sorts of systems, you can decompose the infected subsystem from the whole system. And then you're asking, really, does the infected subsystem grow, meaning do the total number of infected, in this case genomes, grow over time or not? With respect to the extracellular component, even if that virus can kill the cell and make a lot of virus particles, does not mean it's going to proliferate. Because if its decay rate is too high, or there are too few susceptible cells available, then its chance of reproducing and proliferating go to zero, right? Because it needs to complete a life cycle. If it gets out and has to find a new host, if either dies before it finds a new host or new hosts aren't available, that means it hasn't completed a life cycle, which means it's not going to be successful at reproducing. Over lunch, Paul and I were talking, and Paul raised the point that not dying is comparable to reproducing. So there's quite a lot of interest in evolution of this trait, this decay rate or degradation rate, and there can sometimes be trade-offs between making a very stable, producing a very stable virus particle versus one that maybe is less stable. Does that actually mean that it can infect more things? If it's so stable that, remember, these virus particles are themselves metastable, they have to have a conformational change so they can inject genetic material. If you make them perfect so that they can never change, they're also not going to be very effective vehicles at infecting new cells. Okay? So if I were to answer a question about how does the extracellular phase relate to fitness from the individual scale, I would say there it is on the bottom. When M gets too high, this is not going to be greater than 1. I could also relate it to Malthusian fitness in the sense of growth rates. When this number gets too high, this becomes less than 0. So the extracellular phase, the mortality or decay rate of the viruses, is critical to understanding whether or not they invade or proliferate. Okay?
So you talked about how mutations might or might not depend on selection. And if you have a system where they can and cannot depend on selection, that produces noise in your data, and how do you work around that when they depend and when they don't depend?
Okay, I'm not sure I quite understand the question, but let me just try to back up and go back to the very beginning sets of the slides, and then if I didn't answer your question, you can ask again. So the systems that historically have been worked on were these virulent phage, in which the resistant mechanisms were due to a change usually in some kind of surface property or maybe even in the periplasm, but in other words, some way in which the virus no longer can successfully absorb, infect, get in, and begin its life cycle. There are a set of systems, I don't know if Kurt, you're going to talk about CRISPR, it's on immunity, but I don't think you're talking about CRISPR, right? You're talking about human immune systems. So CRISPR stands for Clustered Regular Interspace Palindromic Repeats, which is now a big biotech business and now getting people in trouble because they've, you've probably heard about this story at Sustec in Hong Kong, where people have used, essentially changed this natural adaptive immune system in bacteria and archaea as a means to genetically modify organisms, and that's being done all the time, including food products. If you've had yogurt recently, you're probably having a CRISPR-fied yogurt, but they tried to do it with humans and babies in the germline, totally different story. The reason I'm saying the word CRISPR is that it's a system in which a virus gets into a host cell, injects it, when it gets in, the genetic material gets in. The host cell recognizes it as foreign, though, anyway, that's itself a complicated piece. Once it recognizes it as foreign, it chops up the virus. Those virus parts get integrated into the genome of the archaeum or the bacteria. They get integrated into this series of cassettes, which then get expressed in terms of messenger RNA, get presented as now detectors. So they've survived the experience, and now their offspring are immune to the thing that didn't kill them. So this is really a form of Lamarckian evolution. If I start to lift weights, my children will be stronger, right? Here it survived an infection, integrated a memory of that infection in its genome heritably so that the daughter offspring have the ability. I'll go back now to the Lori-Delbert case. If they had used something like Streptococcus thermophilus and associated phage, their immune system has to do with that kind of adaptive immune system, in which case it requires the interaction. There's no way for that preliminary or pre-selection mutation event because it hasn't interacted with the virus. So it really is mutation tied to selection rather than mutation independent of selection, and in those kind of cases, you expect small fluctuations, highly repeatable outcomes. Though there is the probability of acquiring that immunity is quite low. Categorically speaking, it's the difference between Poisson-like binomial fluctuations, where things are very repeatable, and these large fluctuation events. Now, I think what you're trying to ask, what happens if cells have both kinds, resistance, and there's a lot of debate about when is a CRISPR-based immune system preferable to a surface resistance-based immune system? I don't have a full answer to that. And at the moment, I'd say people are more interested in the evolution of the systems rather than how would it have affected the Luria-Delbert interpretation. That was a long answer, but I think I got to where you wanted to go.
Other questions?
Someone new? Yeah, go ahead.
Mine is more like biological wondering.
Yep,
I
go
was
ahead.
thinking about, is there possible that more than one virus invades a healthy cell?
Yes.
If it is, does it make a difference, like if the invasion, the disease, has a really long latent period?
So now you're asking these are good biological wonders. So there's two facets to the reasons why it could be interesting to think about multiple infections. First of all, it could be multiple viruses of the same type, and it turns out the answers are different in terms of outcome when one or more virus of the same kind gets into a cell. The classic example is phage lambda. I'll talk more about that on Friday. So that's multiple infections. There can also be co-infections in which you have different kinds of viruses with often very different strategies. And part of the reason my group is becoming more interested in latency is we're finding more and more evidence of co-infections being common in natural systems. So I can decide whether or not maybe I'll add a few slides to Friday, but we've worked in these archaeovirus systems in which we look inside cells. These are SAGs, so we can do genomics and try to see who's there, and we find that most of the cells are infected by more than one kind of virus. And so we don't quite know what that means, but you can imagine that if it makes an interesting game theory, because if two viruses are inside, if one decides to burst earlier, you might think that would be the winner. But if the environment outside is terrible, even if it does that, it's not going to benefit. So does it mean that there has to be some spatial structure or other means in which these viruses are communicating with each other? And I just want to point out that viral communication is not impossible inside cells. They're regulating what they do via gene regulatory feedback. And so if they share some common gene regulatory components, certainly multiple infections they do, right? So if you have multiple viruses, each one of them you can think of as a bit of a genetic program, but they share a common pool to some extent, right? So there can be coupled decisions as well. So I don't have a full answer, but I just want to make sure the co-infection and the multiple infection story are both interesting, but they're a little bit different.
They must be coupled to invade?
No, meaning just once they're inside, they don't have to be, no. Once they're inside, what I mean is that, I'll talk more about the phage lambda system on Friday. Viral genome gets inside. We have two viral genomes. If these are the same viral genome, they're expressing messenger RNA and proteins that could bind and have feedback to both. So once they're inside, they become a coupled system. And because these systems are nonlinear, it turns out there can be different kinds of outcomes depending if one or two or more infect.
But invasion is a rare phenomenon.
Infection is rare. Again, I can just tell you what we've seen, that it depends on densities, right? When we look at these archaeal systems, and also we're finding more and more from there are a lot of viruses that are already in the genome when a new virus shows up. So both because they're already chronically infected or latently infected, I'm no longer so convinced that infection is such a rare event. Maybe two active infections, because that's shorter, but there can be a chronic and an active, and so on.
Are there some mechanism to make M negative?
M negative. So here, the only way for the viruses to reproduce, going back to obligate intracellular parasites, is that they need to find a host. So I tend to partition my decay as a negative term. I was actually going to, if I were going a slightly different direction, could I make M a function of V? Meaning, is it really so that viruses have a characteristic lifespan, or do they age? As soon as I make M a function of V, or some other internal state variable, it may be that they stick to each other and become inactive, or they have internal configurations that we don't quite understand. And that's quite interesting, because once this is not just an exponential decay, it opens the possibility for long duration times, and that really changes the story. But for the most part, I keep that negative because that's my decay term, and I put the reproduction on the positive side.
It may exist. I mean, it's not in your model, but outside it may.
Well, so my point is that the positive contributions to virus particles, if I had a positive term, that would mean that virus particles can make more virus particles. I don't know any mechanism by which that's so. Get inside cells, that's where virus particles are produced. Right. There's lots of variation on how they produce those virus particles, depending on the type, but there has to be some interaction with the cell. Yeah.
So you briefly commented on the left column of the Luria-Delbruck experiment, right? That why that might not be so unreasonable, because nowadays it sounds like outdated idea. So maybe could you comment on that? I think you said...
No, it's not that it's outdated, but and this has been said by, I've said it a few times in lectures, and I've seen it read as usually an aside, as sort of an alternative version of history. If they had looked at a different organism, maybe we would have had this other idea in mind. I think both ideas are appropriate. Depends on the resistance mechanism. For the most part, when we think about resistance to viruses by microbes, we think about surface resistance or things that are changes that aren't heritable, in the sense that they don't, excuse me, not heritable, not dependent on selection. They can already be there before you add the virus. Whereas the CRISPR system, because it's an acquired immune system, there's not a sense of what does it mean to have immunity to a virus if you haven't interacted with it. Now, one caveat there, because viral genomes are not entirely distinct, to become immune to virus A, I could interact with virus B, and if virus B, if I pull in part of its genome that virus A shares, then all of a sudden I have gained immunity to new kinds of viruses that I've never seen before, interacted with before, because they share that common short sequence of genome, right? And that short sequence is usually something like 15 to 26 base pairs, something around that length, as part of the CRISPR array.
I have one question. You have stressed many times that all these models, they produce a phase space diagram which is rotating counterclockwise. Is this going to change?
Wednesday. So Wednesday. Yeah, so we're actually, that's been part of the fun, is finding when and showing evidence. No, I have one more. There's evidence that it actually can do different things. Which way it goes. Right, there's not a Coriolis effect where we're going to flush the toilet and see the viruses go down one way or the other. But there is going to be, we're in the southern hemisphere, but it can be both ways in both hemispheres, but I will talk about why that is on Wednesday. Okay. Yeah, one more question.
So the idea that the mutations are previous to the contact with the virus comes from the notion that mutations are random, right? So, like, do you think that if we ever understood, like, if mutations, how mutations are generated could actually change this, how we look at the interaction between the environment and the cell?
Yeah, so there's quite a lot of work, and this is one of the tricky parts of evolutionary biology is trying to make sense of the fact that random mutations and selection can give rise to non-random change, presumably. And so, although it's clear that the rate of mutation, of random mutation, can go up or down, and there can even be some degree of site specificity, but I do not think that there's any evidence of directed mutations. We could think of selection as providing some direction given the selected background, but the mutations, whether they're at one level or another, we should think of them as random.
So, like one of the two randomness, like, because you hear that randomness is like in quantitative, in quantum theory, right? Well, so you say that randomness.
Can I interrupt just there, because I don't want to invoke quantum, I don't think we need quantum theory to talk about the randomness here. I think we just need a small probability at a site per replication.
No, I don't mean that we need quantum theory, but when you think, well, when you ask people, like, where is randomness in nature, they always refer to well, at the quantum level there is randomness, but Dougal said that at the mutation level there is actual, like, real randomness, not just unknown.
First of all, you're talking to different people than most people are talking to, because when you talk to people about randomness, most people don't say quantum theory.
Yeah.
I guess certain people might. The randomness I'm talking about now is that this is a biological machine that does not have error-free replication.
Okay.
So sometimes in the process, it will incorporate a base that is not the base that is an exact complementary match to the other strand, or if it's single strand, it's not making the exact base. There's a probability these things happen. There's error correction mechanisms that themselves can change. So we should think of it as the incorrect incorporation of a mismatch base, which happens at a frequency, and in that sense, that's what I mean by random.
Okay.
And that is the mutation. Yeah. Okay.
One last question, or?
Okay, one more. And coffee.
Sorry, I don't know if I quite understood the basic reproduction when is equal to 1, because I understood that there is the point when we have like coexistency. So I wonder if the conditions of the system is like how in the terms of biologically is that point.
Yeah, so I should be careful because there's in the mathematics and then the system.
Yeah.
Okay. Mathematically, these systems, and I haven't given all of it, but your question allows me to raise it. Mathematically, these systems, depending on some of the life history traits, imply that if this were operating just like these mathematical systems, it would go circle around and get to a fixed point. And that's what I mean by this coexistence point. And at this point, at that density level, there's a corresponding V star, then the effective reproduction number is 1. But there's not another, there's another color. But it's also possible, as I said, there were, you can't see, that's insane. Anyway, let me just draw it bigger. It's okay. Yellow, you can, anyway, we're getting late here. If there's coexistence in a limit cycle, then you can see when we're down here, as it's growing, it's making more virus. So it's oscillating between +1 and less than 1. On average, it's going to be 1 over the entire orbit. Biologically, these oscillations, which are large, are more similar to those scenarios.
Okay.
Yeah.
Thanks.
Yep. Okay. Thanks, everyone.
Okay. So we are back here at 5, okay? 5 o'clock.
I have a doubt, actually.